This is part 2 of the Chapter 8 lecture notes. We had left off, we were talking about 3 of the different neurons as far as differences in structure. So the one we're most familiar with is the multipolar neuron, and it's got multiple dendrites branching off of the cell body, and then one single axon that carries the action potential away from the cell body. The dendrites receive the sensory information, and the axon carries the action potential, or we also call the action potential a nerve impulse, but it carries that away from the cell body. In the case of Chapter 7, we saw that action potential, once it arrived at the axon terminals, which are these structures here, the axon terminals were synapsed with a motor end plate of a skeletal muscle. And so we call that the neuromuscular junction. So we definitely see multipolar neurons that are sending signals to skeletal muscles, and we see multipolar neurons in the central nervous system. Unipolar neurons are the cell body is off to the side. It's not connected directly to the dendrites and the axon. And then you have a single fiber. The dendrite is the part before the cell body, and the axon is after the cell body. But it's a single fiber. In other words, the dendrite and the axon are continuous with one another. And then the bipolar neuron is very similar, except the cell body is in the middle between the single dendrite and the single axon. So here you see, images of those, excuse me, of those 3 different types of neurons. And now let me find where we left off. Okay, and this is the last slide that we were on. Oh, this bugs me when this happens. I'm sorry. Hold on. I'll try to do it fast. Okay, we're just going to have to deal with it. I don't want to have to, I've restarted this a couple of times because I'm having problems with my laptop. But anyway, let's just go through this information. So we have sensory neurons or afferent neurons, and they receive information from sensory receptors. Like if you were to touch something hot, you have sensory receptors in your skin that are sensitive to heat, and they would send that. They would connect with a sensory neuron, and they would send that information to the sensory neuron, which would send that information to your central nervous system. Motor neurons or efferent neurons carry instructions away from the central nervous system to effectors, and effectors are going to be responsible for the response to this information. Interneurons or association neurons are located entirely within the central nervous system, and their job is to integrate the information between the sensory and the motor neurons. Okay, so a little bit of information about sensory neurons. There's about 10 million of them. The sensory receptors that send information to the sensory neurons can be somatic sensory receptors, and within the somatic sensory receptors, we have external receptors that monitor the external environment. So examples are going to be pain, touch, pressure, pressure reception in the skin, vision, hearing, and actually other special senses like taste and smell, okay? And then proprioceptors are going to monitor position and movement of the body. So there are proprioceptors, for example, within the skeletal muscles and joints. And so these receptors will be connected to sensory neurons, and then that information will be sent to the central nervous system. But there are also sensory receptors that are connected to our viscera, which is our internal organs. So visceral receptors or internal receptors monitor the activities of internal organs and provide sensations of distension, like within the stomach or bladder, deep pressure and pain. And motor neurons are the neurons that lead away from the central nervous system and send information to effectors so that there will be a response to that sensory information. There's about 500,000 motor neurons. If they are somatic motor neurons, that means that they stimulate or innervate skeletal muscle. If they're visceral motor neurons, that means they innervate cardiac muscle, smooth muscle, and glands. And then we have interneurons. There's about 20 billion of these, and they're located in the brain and the spinal cord. They're the links between sensory and motor neurons, and they also play a role in higher functions. So that's neurons. The neurons are, remember, neuron is just another word for nerve cell. They're the actual nerve cells. And then there are cells that support the neurons, and they're called neuroglia. Within the central nervous system, we have 4, the astrocytes, oligodendrocytes, I'll say that again, oligodendrocytes, microglia, and ependymal cells. Within the peripheral nervous system, we still have the microglia and ependymal cells. No, we don't. Okay, I'm sorry. There's only the satellite cells and the Schwann cells. Okay, so the satellite cells in the peripheral nervous system are analogous to the astrocytes in the central nervous system, and the Schwann cells in the peripheral nervous system are analogous to the oligodendrocytes in the central nervous system. So let me see if I can get this. It's still not doing a presenter view. All right, I'm going to try this again because I want you to be able to see it bigger. All right, let me try this one more time. Now it'll let me do it. So this image is showing you the different neuroglia within the central nervous system. Okay, so ependymal cells, which are right here, the ependymal cells are cuboidal epithelial cells. They're simple cuboidal epithelial cells, so that there's an example of where you would see that tissue type. And they line the fluid-filled passageways of the central nervous system. Microglial cells are phagocytes, so they help remove pathogens. They remove anything that could harm the nervous tissue in the central nervous system. And then the astrocytes form what's called the blood-brain barrier. Notice that they have all these extensions, and the extensions wrap around the blood vessel, and they wrap around the nerve fiber. And what that does is form a barrier, so that because what's in the blood can be dirty, it can be harmful. The blood can carry poisons, toxins, pathogens, all kinds of harmful things. And once our nerves get harmed, we just learned that they're not capable of dividing, or that it's very difficult for them to divide and repair. So the astrocytes form that blood-brain barrier that helps protect our neurons and our nerve fibers. And the oligodendrocytes form wrappings or myelin around some axons. And that myelin causes the action potential or the nerve impulse to travel much faster down those axons than without the myelin. So in the central nervous system, oligodendrocytes form myelin or wrappings around the axons. And here we have astrocytes form the blood-brain barrier. Oligodendrocytes form myelin in the central nervous system. And now we can talk a little bit more about white matter and gray matter in the central nervous system. White matter consists of myelinated axons because myelin appears white. It's filled with lipids and it appears white. And gray matter consists of unmyelinated axons and cell bodies that contain those Niesel bodies that we talked about. So that they all have the gray color. So that's where gray matter and white matter come from. Microglia are phagocytes, so they play a role in immunity and defense. And ependymal cells are the cells that produce and circulate CSF, which stands for cerebrospinal fluid. Now, the peripheral nervous system, instead of oligodendrocytes, it contains neuroglial cells called Schwann cells, and what they do is form the myelin that wraps around the axons in the peripheral nervous system. And satellite cells have a similar function as astrocytes, except you find them in the peripheral nervous system instead of in the central nervous system. So they form a barrier between blood and the neurons. Okay, you may see the term ganglia, neuron cell bodies that are collections of neuron cell bodies in the peripheral nervous system are called ganglia. Bundles of axons are called nerves. So when we learn the names of all these nerves, that they are actually bundles of axons. An axon is a nerve fiber, and then we have nerves, which are bundles of those nerve fibers. We have two categories of nerves, spinal nerves and cranial nerves. And I'm going to skip this. Name the structural components of a typical neuron. So be able to name and describe dendrites, cell bodies, and axons. Examination of a tissue sample reveals unipolar neurons. Are these more likely to be sensory or motor? And the unipolar neurons, remember, were sensory. If we go back in the slide just real quick, they are, it just says here, most sensory neurons are unipolar. Okay, go back to our questions. Identify the neuroglia of the central nervous system. So make sure you can describe, name and define the astrocytes, oligodendrocytes, microglia, ependymal cells, satellite cells, and Schwann cells. There were 6 of them, okay? And make sure you know if they're found in the central nervous system or the peripheral nervous system. All right, which type of glial cell would increase in number in the brain tissue of a person with a central nervous system infection? And that would be microglia or microglial cell. In the peripheral nervous system, neuron cell bodies are located in ganglia and surrounded by neuroglial cells called satellite cells. All right. Now, we're going to talk about action potentials later, but first we have to understand something called the resting potential. The resting potential, if we were talking about this in physics class or physical science, we would call it the voltage. The resting potential is actually the voltage across the plasma membrane of any cell, but the resting membrane potential of neurons is negative 70 millivolts. So that means that the inside of the cell is negative relative to the outside. So it's more negative than the outside of the cell. And that resting potential is necessary because when an action potential occurs, it depends on changing that voltage to a more positive value, okay? So the resting membrane potential is negative 70 millivolts in a neuron. And these are some factors that influence that negative 70 millivolt value, okay? The one thing is the sodium-potassium pump. The sodium potassium pump pumps 3 sodium ions out